What I'm going to do now is try and give an overview of some of the model-based processes that go into these calculations of emissions. Hopefully it'll give a bit of a background to those of you who don't do this kind of biogeophysical, biogeochemical modeling. For those of you who do, it might be a bit going over old ground, but we'll see. So just to put this plot here up again that you've seen before, I'm not going to go through it because Joe went quite nicely through most of the sinks and sources that really matter here. One of the things I will highlight is that the GPP, the total uptake of carbon by the terrestrial biosphere, is we reckon it's about 120 petagrams of carbon per year. And when you compare this to the amount of carbon in the atmosphere, that implies that every 7 years, the carbon in the atmosphere is being pulled through the biosphere. Now, that doesn't mean it's being removed by the biosphere. Much of it goes straight back out into the atmosphere through respiration. But it gives you a kind of idea of just how much of an effect and how closely linked these different parts of the carbon cycle are. This is, again, the equation that Joe showed you earlier. So we do these carbon budget calculations, and we can say that the change of the CO2 in the atmosphere is simply a function of the emissions from fossil fuels and land use change we put in, with the ocean, the land sink taken away from that. And of course, what we're interested in here is the land sink and the land use change emissions. However, I mean, these are really quite artificially separated when we think about this. When we actually think about the processes involved, they're really closely linked, and it's very difficult to separate the land use change emission from this residual sink. And as Joe mentioned, there's parts of this that we know really quite well, and the bit that we're interested in we really don't have a good handle on at the moment. So I'll just start by going through the basic elements of the terrestrial carbon cycle, a couple of definitions which are easy to get confused. So obviously we have the uptake of CO2 by the plants. This is photosynthesis. And then some of this CO2 is quite rapidly returned to the atmosphere through the plant respiration. Then these plants, so when we term that photosynthesis gross primary productivity, so one of the definitions. And the net of the plant respiration and the photosynthesis, we term the net primary productivity, which are two terms you'll hear a lot in talking about the biosphere. And then, of course, these plants, they die or they turn over their leaves and they create litter, and this litter is broken down and it respired, and this is a soil respiration flux. And so when we look at the difference between these fluxes, that gives us our net ecosystem exchange, or the net exchange of carbon between this small part of the ecosystem and the atmosphere. And this is typically what we'd measure with a flux tower, for instance. But of course, there's other processes that happen on a much longer time scale. So fires return CO2 to the atmosphere, and fires don't generally happen every year in most ecosystems. So in order to take the full carbon accounting of an ecosystem, we have to take into account these things that happen a bit less regularly, or interventions by humans in the system, for instance, through harvest. And we term the sum of all of these different fluxes in and out of the terrestrial carbon system the net biospheric productivity. So this is the net amount of carbon taken up by the whole biosphere. And you'll see both of these terms in the literature, and sometimes you see them confused, but it actually makes quite a difference in the net flux that you're looking at, whether or not you include fire. The fire flux is maybe a couple of petagrams a year, and when the net flux is 2 petagrams a year, you see it's important to be clear on the definition. So how do we go about understanding this? Well, we have several different sources of measurements that we can use to try and understand the carbon cycle. We have flux towers, where you can make direct measurements of the fluxes in and out of the ecosystem. Particularly, we can make direct measurements of GPP and NEE. We can also make measurements of the total respiration, but here we start having to make some assumptions. Flux towers have a limitation that they're only limited to a spatial coverage. It's essentially the footprint of the tower, maybe a few hundred meters in each direction, depending on how high the tower is. But they have a very high frequency, which can tell us quite a lot about aspects of the system and how it works. On a completely different scale, we have satellite measurements. So we have, for instance, a normalized differential vegetation index. It's essentially an index of greenness. We also have now LiDAR measurements of canopy height, which we can use to derive biomass. And we can also, to some extent, derive GPP, but this is generally derived as a secondary product from other measurements. So the trouble with satellites is we're generally measuring something that's a change in a radiation. So we can get a column measurement of how much of a gas we have in the atmosphere, or we can get a change in the surface energy balance in various different wavelengths. But in order to turn those into the fluxes that we're really interested in biosphere, we often have to make a level of assumptions or use models or calibrate them against surface measurements. And so they're a less direct way of extending the knowledge that we already have of the surface, maybe. And of course, these have a much lower frequency than flux towers. And then we have another class of measurements, which is inventories. So we go out and take, well, I don't go out, but some people go out and take measurements of the biomass in forest, how fast the trees grow, the different allometries, the different allocations of carbon to different parts, whether it's the roots, the trees, the trunks, etc. And these have generally an even lower frequency because it takes a heck of a long time to go around a forest and sample all the trees in it. But basically, this isn't an exhaustive view of all the measurements we have, but it's kind of to point out that we don't have any nice global scale overview of the key fluxes that we're really interested in trying to understand the global carbon cycle from the terrestrial point of view. And so what we have to do is fill in the gaps with process-based models, or we have to fill in the gaps with models, and we tend to use process-based ones. So the models that we use today have evolved from a variety of different backgrounds. Just on the left side here, I go through some of them. So we have the GCM land surface models, which have been put together to give the boundary between the atmosphere and the land surface for global circulation simulations. And this is really focusing on the surface energy balance and the surface roughness. Then we have the terrestrial biogeochemistry models, really looking at the exchanges of the trace gases, particularly CO2 with the surface and the atmosphere. Another class going back in time is the plant geography models. So what kind of plants grow where? Are there bioclimatic limits that say particular plants grow in these regions, for instance? And then we have vegetation dynamics. So how does different types of vegetation interact with each other? What are the mortality processes, and how do they affect the kinds of vegetation we have? How is it established? And so over the last 20 or 30 years, these different fields that were once separate have now started to merge quite a lot as we've got an appreciation of how the Earth system, everything is connected and interacts with each other. And so I've tried to group together the models that we generally use today into three different types. We have the land surface model again, but this time as part of an Earth system model. So this is incorporating both energy balance and biogeochemistry. And the basic version of that uses a static vegetation, and this is commonly what's used in CMIT5 models that we use in the last assessment report. Coming from the other direction, we have the dynamic global vegetation models. And these are focusing on being able to simulate the type of vegetation that we would have in a particular place under a particular set of environmental conditions, and then the exchanges of the various gases that go along with that. And the increasing trend is to start incorporating DGVMs into land surface models, so we have this combined class which includes both of them. One of the real problems with modeling the terrestrial biosphere is the range of scales we have to cover. So we have a huge range of spatial scales. We have to go from the molecule in terms of photosynthesis to scaling this up to a leaf. And then the leaf is obviously just one part of a tree. And the tree is part of an ecosystem that may have a whole range of different trees and grasses and completely different to just multiplying one tree after another. And then within the grid cell that we typically model at, which is anything from 0.5 degree to 2 degrees across, then we might not just have a forest, but you have all different types of land cover. So we have an increase in heterogeneity, and of course this happens again at the globe. So as the scale increases, the level of heterogeneity that we have to incorporate increases, and it's not just a simple case of saying, okay, at the globe we have 12 billion leaves, so if we model 12 billion leaves, we'll get the right answer for GPP. There's so many nonlinearities in there that it just doesn't work that way. And with these differences in scales, we also get differences in time scale. So we have processes that we really need to model at very high time frequency, so the biogeophysics in terms of minutes or maybe half an hour, depending on the model we're using, and its interactions with the atmosphere needs to be a very high frequency. Similarly, emissions of some compounds, such as volatile organics, need to be a relatively high frequency, at least an hour or so. Then we have things like phenology, so the turnover of leaves. Here we're talking days to weeks is probably sufficient to capture the seasonal cycle in leaves. And then there's other processes that we can really just treat annually. So vegetation dynamics and soil biogeochemistry, we can often get away with making simplifications, so we only have to run those parts of these models annually. And in DGVMs, we actually tend to do these processes daily rather than sub-daily, just to make them a bit lighter and easier to run. So really the point of this talk is to go through some of the massive number of assumptions and uncertainties that we have in putting these processes together. And really there's enough here to fill an entire summer school. And there are whole training schools just dedicated to this subject. So I'm really going to skip over lots of things and miss many things out. But if you have questions, then I'll try and answer. So I'm going to go through these aspects of the system. I won't read them out to you. You can read them and we'll get to them in a few minutes. So first off, we have to run our models with something. And generally we run these models with boundary conditions that are mostly atmospheric. So all models we're looking at the terrestrial biosphere, we have to provide them with shortwave radiation. We also need to provide precipitation measurements, and the atmospheric CO2 mixing ratio is also very important. And then, depending on the model type, we either need the incoming long-wave radiation so that we can make a calculation of surface energy balance, or we need the surface temperature directly, which we can take from a different model. And then, depending on the different model types, some also need humidity, wind speed, deposition of different biogeochemical components. Aside from this, there's also non-atmospheric parameters relating to the soils in particular, but if you have static vegetation, you might need a vegetation type map. If you're starting to consider land cover, then you need land cover fractions or different management variables. How much nitrogen you're using to fertilize croplands, for instance, might become relevant. And generally, we either get these from historical reconstructions where they're available, we have historical reanalyses for climate that we can use, or we take them from global climate models if we're running offline, or we give assumed emission trajectories based on the scenarios that Mark was describing yesterday and put these into our Earth system models. So going through the processes, I'll start with photosynthesis. I think you all know that photosynthesis is the combination of CO2 and water to make some sugars and gives out oxygen. In most of these models, we use a very standard way now of calculating photosynthesis, which is known as the Farquhar model, which comes from the early 1980s. It's a very neat process-based approach of trying to capture the main aspects of photosynthesis from our point of view. And nearly all the major models that we're looking at nowadays either use this model or a derivative thereof. And essentially this model comes down to a minimum of two different rates which limit the rate of carbon assimilation. One is the regeneration of ribulose biphosphate, which you can essentially consider as the rate of energy supply from light into the system. And another is the carboxylation capacity, which is the rate at which the enzyme Rubisco can take CO2 and stick it into a larger organic molecule. And so we simply take the minimum of this as our these two rates within this equation as our assimilation rate. So this assimilation obviously depends on the amount of light we have. You can see in this light response curve down here that as the photon flux density increases, then our assimilation rate also increases until a point where it plateaus out. So at low light levels you get a strong increase, and as light increases relatively high levels, you don't get any more effect. The leaf simply can't deal with any more intake of, it can't handle any more light. can't make use of it. As well as the light level, temperature is also important. So this particular curve this is for one species, so don't take too much note of where the temperature maximum is, but we have an increase of the assimilation rate at low temperatures, reaching some maximum, which is species and environment dependent, and then a tailing off due to mainly the activity of the enzymes involved. So as Rubisco gets warmer, it gets less effective at fixing CO2. It starts to get it confused with oxygen and trying to stick oxygen into the carbon molecule, and that's energetically not very good, and it costs the plant carbon to deal with that. So we get a drop off in the net assimilation rate. Now obviously, because the plant is fixing CO2, the amount of the substrate it has to work with is also important. So for the vast majority of plants globally, so all trees and temperate grasses, we see a very strong response of the assimilation rate to the amount of CO2 that we have in the atmosphere, and this is the CO2 fertilization effect. So as you increase the CO2 concentration, you get a very strong increase in assimilation. The exception to this is plants that go under C4 photosynthesis, which is the tropical grasses basically, and they plateau out in terms of their CO2 concentration at a very low level, which is far below what we have in the atmosphere today. But that's not the whole story, of course. We know that plants are also limited by nutrients. And nitrogen is one nutrient that gives a particular fundamental control on photosynthesis. So this plot here shows the change in the net photosynthesis as you change the leaf nitrogen content. And you can see that there's differences between broadleaf and needleleaf, which I won't go into now, but as you increase the amount of nitrogen, the plant has more capacity to photosynthesize, because it can make the proteins that it requires. Rubisco needs a lot of nitrogen in order to be made. So the amount of CO2 matters, and the amount of CO2 in the atmosphere matters, but what really matters is the amount of CO2 that gets into the leaf. So it's the CO2 inside the leaf that actually controls the rate of photosynthesis. And the plants take in the CO2 through their stomata, small pores on the underside of the leaves, and this is essentially a trade-off, because as you open these pores to take in CO2, you also give out moisture, and plants have to reach some kind of optimum or happy medium state where they're not losing too much water, but they're getting in enough CO2. And this could be quite a difficult process to model if we think about all the biochemical things that are biophysical processes that are going on inside the leaf. But what we tend to do here when we're modeling it is what we do with many environmental problems. We simplify them by saying that the plant does what's optimal, and therefore we can bring it down to a very simple equation. Plants don't always do what's optimal, or they probably do what's optimal, but they're taking into account many other things, which make these assumptions a little bit uncertain in many cases. But the way we typically model this exchange of CO2 and moisture is to say that the plants optimize the stomatal conductance so that they get the maximum CO2 intake versus an acceptable amount of water loss. So in the LPJ model, we say that stomatal conductance is a function of a minimum, to which we add a function according to the net assimilation rate, the amount of CO2 in the atmosphere, and then a function which is controlled by how stressed the plant is in terms of the amount of water that it has. And so you can also see here, because there's the atmospheric CO2 concentration is included, that you have not only a direct fertilization of photosynthesis through the CO2 acting on the photosynthetic process, but you also have a change in the water use efficiency through the stomatal conductance. And both these processes are important at the global scale. So what does this look like? If we look at GPP globally, this is some output from LPJ guess. It's just indicative. We have a lot of strong uptake of CO2 in the tropical rainforests. We also have a strong uptake across the the northern mid-latitudes, but less so because here we have to take into account seasonal effects. In the tropics, we have high temperature, we have a lot of water availability, the plants can be productive all year round. Just to ground the model here with some measurements, we can compare it to chlorophyll fluorescence measurements. These are quite a new set of measurements, just become available. When chlorophyll takes up light, shortwave radiation, it also fluoresces a bit in a slightly different wavelength. And now we're able to separate this wavelength of light, which is fluoresced, and it gives an indicator for GPP. And you can see that this indicator here has a very similar spatial layout to the model, with the exception of this little bit in northeast America, which I'll come back to later. So that's production. What about respiration? Respiration we usually do much more simply than we do production. We basically divide plant respiration into growth and maintenance respiration because they have different intrinsic rates. Growth respiration is generally at a higher rate than maintenance respiration. And we model these simply at least in LPJ gas, but this is kind of generally, once again, representative. It's a function of the species, the amount of carbon in the leaf, or the amount of carbon in the tissue, more to the point, and the carbon to nitrogen ratio in the particular tissue. So we have different rates of respiration in leaves versus in wood versus in roots, depending on the amount of nitrogen relative to carbon we have there. And then we modify this by a function that's dependent on temperature. This is the temperature function in the LPJ models, at least the ones that I've been working with. And you see that the temperature, the function increases very strongly with temperature. In reality, there's some kind of maximum, but many models don't take this into account. So as the temperature increases with climate change, for instance, we get a strong increase in respiration in these models. Growth, we do actually much more simply. In our case, we just say it's a fraction of the leftover carbon goes into growth respiration, and completely gloss over all the processes that go into that. So this is how we generally do it, and it's a huge simplification of the underlying processes. And there's, if you're interested particularly in how respiration is modeled, you can look at this paper here, which goes through some of the different decisions you can make in modeling respiration, which we typically don't do at the global scale at the moment, although we're trying to get better at that. There's also things such as acclimatization. So there's quite a lot of evidence if you take a particular plant from one climate and you stick it in another climate, it doesn't just move up this curve, but it actually acclimatizes to that new environment and kind of resets its base rate of respiration. We don't generally take account of that. And similarly, we don't account for how models respond in how plants respond in our models in terms of stress. So there's a lot of evidence that we get respiration going in completely the wrong direction in drought conditions, for instance. Soil respiration, very similar. We take simple lifetime functions, so we generally split the soil into pools of different carbon, depending on how quickly that carbon breaks down, and we characterize each pool by a lifetime. And then we modify that lifetime according to the temperature and the moisture. So here we have exactly the same temperature function that we use in the plant, in actual fact. And then as the amount of moisture increases, we say that the respiration rate increases, which is generally true up until a point when the soil becomes waterlogged. Actually, we don't account for that here. And again, this is a huge simplification. And when you consider that a lot of the response of our biospheric models in terms of carbon going into the future is an increase in respiration in soils, this is something that we really need to look at more closely, and is now being looked at more closely in the modeling community. And there's a whole range of different factors that go into this, and some models now coming out that actually look at how we can model the processes of the actual bacteria in the soil are doing when they break these carbon molecules down. So this is just the net ecosystem flux balance. It looks very similar in terms of the spatial breakdown to the GPP. So we have generally a high uptake in the tropics, and also some uptake in the northern mid-latitudes, but smaller. But this is basically the spatial breakdown of that net terrestrial sink that Joe pointed out earlier. The other interesting aspect, I think, here is the seasonal aspect. You see there's much more land in the northern hemisphere than in the southern hemisphere, and so the vegetation cycles in the northern hemisphere tend to dominate the seasonal cycle of CO2. And you can see this both at measurements from flux sites. Here from a paper used to evaluate LPJ, we have a net outflux of carbon in the winter months and an uptake in the summer. And you can actually see this in the seasonal variation of CO2. You can see the seasonal cycle in the vegetation. The main thing that controls the seasonal variation is the biosphere, as opposed to anthropogenic emissions, which control the overall trend. So that's a really brief run through the main fluxes that we try and simulate. How do we put these all together? We do this using concept of pools, as I mentioned for soil. So there's a pool of carbon in the atmosphere. We take this up through photosynthesis, and it goes into a pool of carbon in the vegetation. And the vegetation dies, or it turns over, and it goes into a set of soil carbon pools, which vary in their lifetime, as I mentioned. And the vegetation pool, we have different ways of representing this and linking it with the actual vegetation we see outside. So it might be that we represent our vegetation through a single average individual, which has characteristic features, or a whole range of different individuals, and this depends on the model we're using. But nearly all of the models that we use at the global scale have some set of allometric rules that decide if that carbon is coming into vegetation, to which part of the vegetation does it go? Does it go to the roots? Does it go to the wood? Does it go to the leaves? And this has obviously huge implications for the carbon, because it changes the typical lifetime of that carbon within the system. The lifetime of carbon in leaves is much shorter than the lifetime of carbon in wood. And then the typical way to scale these up to the grid cell is to have fractional coverages, a tiling of different plant functional types. So we might say that 1/3 of the grid cell is plant functional type 1, and another 20% is this type, and another 25% is this type. So in that way, we can represent different plants in the grid cell, but I mean, you can't look outside the window because we've closed the blinds. But if you could look outside the window and looked at the forest, then you'd see that the forest doesn't separate species into little blocks. You know, everything is mixed in with each other, and this has quite fundamental implications for how the ecosystem functions. So if you consider a forest, a real forest, you have different species and you have different ages of species, and some plants are taller than others and overshadow others and therefore have a competitive advantage for light. Others have larger root networks, and that gives them a competitive advantage in terms of gaining nutrients when nutrients are in short supply. So some models, LPJ guess being one of them, and I'm unashamedly going to show you lots of things from LPJ guess because it's the model I work with, use an approach where you also break down those plant functional types into different age groups, and you mix them together in the grid cell, so that you try and account for some of this different this heterogeneity that you have in natural systems. And if anybody's more interested in how we do that, then I can go into a bit more detail later on offline. Plants also die, of course. It's not just a case of taking in carbon and respiring it, but eventually they die and that carbon is returned to the soil and eventually, in most cases, the atmosphere. And there's a variety of different ways that we represent these death mechanisms in the models. So we might use bioclimatic limits. We say that this particular plant type can only survive between a temperature of 10 and 30 degrees, and if not, then it dies if the temperature stays outside that for a long period of time. Or if the plant can't maintain a positive net primary production for a set period of time, then it dies. Similarly, growth efficiency, this is a minimum amount of biomass increase per unit leaf area. And then there's the simpler things such as a maximum age that a plant can live to, or a set background rate after which plants die. Oh, wrong way. So the thing about these is that all these mechanisms, they're kind of logical, they make sense, but they're not really very well tested. And in particular, they don't really explicitly represent the mechanisms that lead to how plants die. So plants die because they don't have the necessary stores and intake of carbon in order to be able to fulfill their basic functions, or they die because they have a hydraulic failure. They don't have enough water within their tissues in order to be able to maintain their basic functions. Either they fall over or they can't run their metabolism properly. So there's quite a disconnect in the way that we represent processes at the global scale and how they actually happen in reality. And as well as these kind of processes that come from within the plant, there's a lot of processes that come from without that can cause plants to die, and ecosystem disturbances. So fire, wind throw, insect attack, such as we see in a lot of the boreal forests, well, not the boreal, the coniferous forests around here. You see a lot of deaths due to bark beetle attack, and this is really quite a big issue at the moment. And this plot comes from a paper which was published last year in Nature Climate Change. It's the increase in the amount of wood in terms of forest biomass that's been affected by disturbance in Europe over the last 40 years. And you can see there's a huge increase in the amount of disturbed biomass, and an increase in all types, so in wind disturbance, in beetle disturbance, and in forest fires. So there's now some evidence that these disturbance rates are not just important for turnover, but they're also increasing with climate change. And this is a major uncertainty that we don't really have a proper handle on in the system yet. Just to give an idea of the possible magnitude of that, this is some simulations we did with LPJ-GESS. It's the change in vegetation carbon storage as you change the rate of disturbance in the system, basically. So moving to the left on this axis is more disturbance. And this line here shows the tropical forest, the mid-latitudes, and the boreal regions. And you see that if you were to double the rate of disturbance everywhere, like we see in Europe, then you would have a drop in the vegetation carbon storage of a couple of hundred petagrams. So this is something that's comparable to all the emissions we've put into the atmosphere over the last 150 years. This is a really large amount of carbon that's sensitive to changes in disturbances. I touched a little bit earlier on plant functional types. There's a huge range of species out there in reality, and we don't have the computational capacity or indeed the data to parameterize our models in order to be able to represent all these species. If we're looking at just regional scales, for instance Europe, we can get most of the major species, but globally we can't even come close. There's thousands of species out there just in terms of trees. So we break these down into groups that we call plant functional types, and we differentiate these by different traits. So by the type of photosynthesis, by whether it's evergreen or deciduous, whether it's woody or a herbaceous plant, or the bioclimatic limits are typical, but there's all sorts of different ways we can do it. And so we break these down into different classes, and typically in global models we have maybe 5 to 15 different classes of plant functional types, which are tried to capture all the variation in global vegetation. And you can immediately see that there's a huge simplification there, and we can broadly get the general distribution of these plant types, but it raises questions of resilience, and there's a lot of evidence that if you have many different strategies in an ecosystem, many different species following different routes, then that ecosystem is more resilient than if you have one type, as we do in these models. So this is just showing a potential natural vegetation map from LPJ-GESS. So the vegetation that would be there if there was no human influence at all, according to our model processes. And this is an attempt at reconstructing what the potential natural vegetation should be based on observations. So obviously there is no observation of real potential natural vegetation. You saw earlier in the earlier talks just how much of the land surface is strongly affected by human management and land use change. But you can see that the broad features agree. So the tropical forests are quite similar in their extent and also the boreal regions. We even get the Siberian larch forests out here, more or less. These models do a reasonable job of getting the biosphere at least as it is now. Going into the future, that's a completely different question. But of course, we know that the biosphere isn't potential natural vegetation. That's why we're all here. There's a huge amount of management that goes on. But until recently, and until the fifth assessment report, in Earth system models and generally terrestrial biosphere modeling, we only concentrated on potential natural vegetation, and we didn't take into account the huge modifications that humans have made to this system. But these managed systems differ fundamentally in many of these aspects. So in a DGVM, we might represent establishment and mortality processes. We'll represent maybe some fire and competition between different plant types. But if you think of a cropland, what you have is you have a prescribed species, and that species may be fertilized or irrigated to increase its productivity. You don't have to worry about establishment because it's sown on a specific date, and mortality is less relevant, although not obviously completely irrelevant, because you harvest at the end of the year. So the processes involved are really completely different. And so this would suggest that the exchange of trace gases would also be completely different. And of course, that's what we see. So this plot here is once again these chlorophyll measurements of GPP. And you saw earlier this large increase, or this high amount of GPP in the northeastern USA, which the LPJ gas didn't capture in its potential natural vegetation. And the reason is that this particular bit of land is dominated by cropland. And this particular cropland is very heavily, intensively managed, so that you have very high productivity there. And what this paper, which came out last year, showed is that this very high productivity in many of the global croplands, particularly in North America and in India, isn't captured in the terrestrial biosphere models that we're using to model GPP. This is an example of a difference plot here between the data and a couple of different types of models. And there are also, this is a very active bit of research actually. There's a couple of papers that came out in Nature just at the end of last year that showed the effect that the cropland productivity has on the interannual cycle of CO2. Not the interannual, the seasonal cycle of CO2. And they showed that there's been an increase in the amplitude of the seasonal cycle over the last decades, and that a very large proportion of this increase can actually be attributed to the increased productivity in croplands in the northern hemisphere. So these managed agricultural lands are really modifying the exchange of gases. Do you have a question? No, what they measured here was the agricultural intensification, and they calibrated their model to the increase in yields that had been seen, and then ran that for the biogeochemical consequences. And it's not just productivity that changes, though. Also, we see large changes in the soil carbon stocks. So this plot here, this is some output from LPJ gas once again. What I've done here is run natural vegetation and then completely change it over into different land use types. So, and then looked at the change in soil carbon after 10 years, after 50 years, and after 100 years. And if we change natural vegetation into grassland, then as a global mean value, this is just a typical indication for the global average response, you actually get an increase in soil carbon. And this is due to some changes mainly in how the allocation and turnover happens within the system. But once you start grazing that grassland, then you probably come out with some kind of more equal response. The soil carbon response as an average isn't particularly different, although there's a huge spatial variability. Some places show us an increase, some places show a loss. And of course it depends on the intensity of the grazing. And then if we move to cropland, you see a very strong loss in soil carbon. And this is partially due to the harvest, which takes out a lot of carbon from the system, partially due to processes such as tillage, which increase the respiration rates of the soil carbon. And these model results, just to show, are broadly consistent with meta-analyses of forest to crop and forest to pasture conversions that we see from observations. All right, so that's my whiz-quick run-through of some of the key processes. I want to just try and bring that all together and talk a little bit about what the projections of the global carbon cycle actually look like. So, as always, I'll start with LPJ gas, but I will mention a couple of other models just to be a little bit more even-handed. This plot on the left shows the terrestrial carbon accumulation from 1850 to 2100, following a strong climate change 8.5 scenario from 2006 onwards. And if you concentrate here first on the blue lines, the blue dashed line shows the uptake of carbon you get just due to CO2 fertilization alone. So CO2 fertilization has potentially a huge uptake of carbon effect in these models. If you look at the dashed dot line, this is the effect of climate, and you see a strong decrease in carbon in the ecosystem, in the global ecosystem. And this is primarily due to the effects of temperature on respiration. So at the higher temperatures, we're getting much higher respiration in soils particularly, and lower soil carbon stocks. And when you do both, you get this blue line, which is partially covered here, which is much smaller than the CO2 effect alone, but you can see it's positive and quite large, at least in our model. So we know that more is important than just the carbon. So nutrient limitations are also important, and the red lines here showed us the same set of simulations, but with nitrogen limitation or nitrogen cycling effects included. And you see that, once again, the CO2 still has a positive response on the dashed line, but it's much, much smaller than it was when we didn't consider nitrogen. However, in the other direction, the effect of climate isn't quite so negative, because increased temperatures, they also increase the mineralization of nitrogen in the soil, and so make more nitrogen available for plants to use. So in some areas, the climate actually releases some of the nitrogen limitation. Yeah? Yeah, this includes vegetation dynamics. And actually, this part of this effect that you see here is that in the lower boreal region, you have a relaxation of nitrogen limitation, which leads to trees instead of grasses being able to grow there, and that gives an increase in the amount of carbon that's uptake by the biosphere, and that's purely a function of vegetation dynamics. You wouldn't get it if your model had static vegetation. So in the LPJ-GES model, we get actually a net carbon uptake that's pretty similar to the carbon-only model, but these other symbols here show the difference for a range of other models in the literature which also incorporate nitrogen, and every other model apart from LPJ-GES actually shows a slight decrease, or some sort of decrease in the amount of carbon taken up under climate change. We've got no idea which is right, because they all incorporate different kinds of processes. Some of those models don't have dynamic vegetation, some of them do, but do it in a different way, and it's really uncertain how this is going to happen. Just to say that this plot on the right here, this shows the latitudinal breakdown of these different fluxes, and I think I only want to highlight here that we have much more uptake in the tropics, and the CO2 fertilization effect is much larger in the tropics. And this comes from, it's basically an outcome of the Farquhar model, which shows that if you have higher temperature, then the CO2 fertilization effect is more effective. So this is, in terms of process terms, this makes sense. Unfortunately, we don't have really yet the measurements to actually see if this is really happening in reality, although they're in the process of being made at the moment. Just to go back to the agriculture briefly, this is some simulations we made with, again with LPJ-GES, but looking at the effect of those different agricultural representations I showed you earlier. So this is the change in the net biospheric uptake of carbon. The negative values are uptake. The blue line is if we just have potential natural vegetation running into the future. So this is historical, this is RCP 8.5 climate, RCP 2.6 climate. we just have potential natural vegetation, we have a strong uptake of carbon, as you'd expect, I guess. Once you include land management, then you lose much of this. But how you include that land management is really important. So if you represent your crops as grasslands, like many Earth system models do, then you get, in our LPJGS representation of that, you get this line here. If you harvest those grasslands, which some models do, then you get this line here. And if you include our full crop model, then you get an even larger increase in the emissions that are given out. So this is something that Almut was referring to earlier, that this is another one of these processes, along with consideration of nitrogen, that may lead to having much higher land use change emissions than we've previously considered in the past. And of course, because we have this budget equation that says we know exactly how much the CO2 is increasing in the atmosphere, and we know roughly the sources from fossil fuels and the loss due to ocean sinks, we have a constraint on the total biosphere effect. So if we increase the land use emissions, then we must also be increasing the size of the biospheric sink, and that means we're missing sink processes, or we're not capturing them properly. So even though Joe showed you that plot where the residual sink from the budget method and the DGVMs actually matched up very closely, that could easily be due to a set of missing but compensating processes. And you can miss processes and have them compensate now, but in 50 years under different environmental conditions, that relative balance might completely change and could change the direction of our projections. So, as I promised, I'd broaden away a bit from LPJGS. But this plot here is LPJGS, but run with a lot of different climate models as input. So first, you can see that with one terrestrial biosphere model, but a lot of different climates, depending on the climate input that you're using, you can either have an uptake of carbon or a loss of carbon between 1850 and 2100, just from the changes in the amount of warming and where that warming happens. And then if you also add into that uncertainty in the biospheric model representations, then you can get something like this lower plot, which is the carbon uptake simulated by CMIT5 system models. The grey bars show the range of the LPJGS results in the top plot, and you can see that the range, when you include this extra level of uncertainty, is even larger and also goes in both directions. So when we're considering land use change emissions going into the future, it's very difficult because we have a moving baseline of what the carbon stored in the terrestrial biosphere is, and we don't even have agreement on what direction that baseline's going in. Just want to touch on that nitrogen isn't the only important nutrient. This is some work done in the MPI in Hamburg by Daniel Goll. The top plot shows nitrogen limitation spatially across the globe at the end of this century in a strong climate change scenario, and you see that most of the nitrogen limitation is in the mid latitudes, and there's not so much in the tropics. However, what Daniel did was then add phosphorus cycling into the model and looked at the effects of phosphorus limitation. And once again, there's quite some limitation in the mid-latitude regions, or they're actually in different areas, you notice. But there's also quite a lot of limitation on the uptake of carbon, or the carbon stocks, in the tropics. And it's the tropics that are really the driver of most of the global carbon uptake in these models. And yet basically all the models but this one have neglected the effect of phosphorus cycling. And there is a lot of evidence that suggests that phosphorus could be the limiting nutrient in the tropics. And therefore maybe our projections for the tropical forest uptake due to CO2 are too positive. Once again, we don't have a good handle on this, just like we don't really have a good handle on anything, it seems. That's not really true, but there's many things that we don't have a good handle on. As well as models varying in their total change as a global average, they also vary a lot spatially. So these are some results from an intercomparison of DGMs made by Stephen Sitch and colleagues a few years ago. This is the change between 1860 and 2099 in the tree cover fraction between 4 different dynamic vegetation models. And you can see that the changes and the direction of changes and the position of those changes is actually, it varies massively between the models. LPJ shows a loss of tree cover in the mid latitudes, which Trifid isn't showing. It's actually showing an increase. And so when you start to look at spatial effects, the models are the uncertainty between the models is actually even greater than when you just look at the global averages. And you see the same kind of thing in the vegetation carbon stocks in these plots here. The other thing I'd just briefly highlight is this issue of Amazon dieback, which resulted from a paper in the early 2000s by Peter Cox and colleagues at the Met Office in the UK, and they ran the Hadley Centre coupled climate model, and they simulated almost complete loss of the Amazon rainforest by 2100, which was quite unexpected and made quite a shock in the community when it came out. There's been a lot of work on this following on to see if this actually happens. Basically, no other model has really been able to replicate the extent of this Amazonian dieback, and indeed the new version of the Hadley Centre model also doesn't replicate it. It looks like it might be a blip. But having said that, if you look at the results from this DGVM comparison, you do see that so the Trifid model here is the dynamic vegetation model and the Hadley Centre climate model, and you see this massive loss basically of tree cover. The other models, they don't show this same massive dieback, but they do show some tree cover loss. So there is some kind of agreement that, or at least a strong possibility that while we're unlikely to have complete loss of these forests, maybe there is some vulnerability of the forests as we see them now. And you see this also reflected in the carbon stocks, which sometimes respond much more strongly than the tree cover fractions do. And the other side of the story, there was a paper a couple of years ago by Chris Huntingford, and also working with the guys at the Met Office. They ran the Met Office dynamic vegetation model, but this time ran it with a whole range of different different climate inputs. And they found that nearly every single model that they drove it with resulted in an increase in vegetation in the tropics, apart from their own in-house UK Met Office model, which showed a decrease. And so the outcome of this paper was they suggested that actually the tropical forests are pretty robust to changes in climate. However, if I go back to the mortality mechanisms that I mentioned earlier, these models aren't necessarily capturing the real mortality mechanisms. There was a paper a couple of years ago that showed that the trees in the Amazon have a very low safety margin for hydraulic conductivity. So basically they live right at the maximum limits that the environment allows them to live at, and you only have to push them very slightly before they start getting hydraulic failure. And so there are questions over whether we represent these processes related to mortality properly. And this is my final slide. Just one thing I think is really important to bring up is that photosynthesis and respiration, the basic rates can respond quite rapidly to changes in climate, but in the vegetation and the biosphere system, there's a lot of lag. So the vegetation dynamics and the soil carbon stocks, the vegetation carbon stocks, these respond on a much longer time scale than the changes in climate. So this is some simulations we ran where we ran the model LPJ-GES with an RCP 8.5 climate change up to 2100, and then at 2100 we fixed the climate, and we waited to see how long it would take for the biosphere to come back into equilibrium and what effect that would have. So you see the time for the biosphere in terms of the net biospheric productivity to come back into equilibrium with the climate is about 200 years after we fixed forcing. And in this time we have a large increase in our vegetation carbon stock and quite a substantial decrease in our soil carbon stock. So while we can look at changes, we can take 2050 or 2100 and say our model projects that we have this loss of vegetation with this climate at this time. This isn't necessarily the loss of vegetation that is truly associated with this climate if you allow the system to come into equilibrium. And indeed, if you run our models now, just fix them at 2005 climate, for instance, and run them into the future, you see there's a large change just from the environmental changes we've had already that's built into the system. Even if we could completely stop all climate change right now, we'd still have large changes in the vegetation. So just a summary. So we have a lack of appropriate observations that mean we have to use models to represent the carbon cycle. We try to capture the key processes, but these are highly simplified ways we go about doing this. And whilst most models predict a strong uptake of carbon by the biosphere under environmental change, there's a lot of uncertainty. They don't universally agree, and different models represent different processes, and there's some processes that are almost completely missing. And so, as you're going to see, or as you have seen from Joe's talk, actually, now we swap them around. These changes in the natural vegetation are completely changing the baseline that we consider when we consider the effects of land use change, particularly in terms of that missing sink that Joe was talking about. And I'll leave it at that. I'll flick through some references just for the copyright.